This Friday, March 17th, you will be starting your lab on human digestion. And I am coming to you with some step-by-step instructions, and I'm going to show you guys how to complete this lab. It is not the easiest lab to work through, so I wanted to give you a visual of what you will be doing and talk you through the lab. If you have questions beyond this help video and the help session that I run on Friday, please feel free to email me so that you can work through this process. So I'm going to start off very basic, and then I'm going to get into the lab in just a moment. But here are the step-by-step instructions for that lab. Here are the objectives. You're going to be able to look at that human digestion and know those functions, how that the digestive enzymes are going to work on the food that we eat. So first of all, the first thing you need to do is read the lab. There are two different areas you can actually enter into this lab. 3.13 also has this, and it says human digestion actions number 1. 3.15 is human digestion actions number 2. They are actually going to be pretty much the same thing. So the only thing that's going to be different is the information that is in this lab right here. You'll probably use mostly 315, but the information here will be different. 313 and 315 will have different information. So you're going to want to start with 313, and then move on to 315. But the assignment is going to be the same. There's not two different assignments. It's the same assignment. So read the introduction of both of these labs, 313 and 315. Make sure you read the lessons. So this is lesson 3.13. Also read 315 as well. Write down notes. That will be helping you in finishing your lab when you get after you get done with your whole lab, and you go back in to answer those questions that are on that lab report. As you're reading that lesson, take notes on the pH. What does it mean for it to be acidic? What does it mean to be basic? What do these colors mean? So make sure that you definitely look at that information. You will be using pH strips to help you identify color, number, and whether that solution is going to be basic, acidic, and or neutral. Once you are in that lab you've read through, you will want this study guide, the student guide. This student guide will go through step-by-step instructions of what must be done in the lab. If you don't have these, your lab will make absolutely no sense. This one says lesson introduction, human digestion actions number 2. You can find the same student guide in the unit lesson 13 as well. Make sure you also download your lab report, because as you are going through the first part of the lab, you will be filling out a chart that is on that lab report. So download that so you have that. Make sure you save it with your name in the title somewhere. So number 1, it'll be easily found, and number 2, when I grade it, I know who I'm grading. And then you will enter into that human digestion virtual lab that will be in this area right here, investigation, human digestion actions number 2. Once you get in there, you're going to view the tutorial. That tutorial will walk you through step by step on a few parts, and then it'll ask you to begin the lab. and start all over again, but that will help you maneuver around in the lab. When you are looking at the directions, this is the student guide. This thing right here, let's go back to it. This student guide right here is going to be looking like this. You are going to do part A. Notice where it's crossed out in red. I will talk you through this. But you are actually going to skip a part in this lab. We're only going to make you do part of the lab. So when you finish the tutorial, it says reset the lab, begin the lab button, and then continue to part B. You are actually not going to do part B. You are just going to move on. So you're going to go from part A to part C. So all of this milk digestion, this is going to be skipped. When you get to part C, this is going to be a little bit confusing. It says to mince the egg. I will show you what that means. You're going to spoon it in. And then this says fill each test tube with 2.5 milliliters or 1 dropper full. That is actually incorrect. You're going to use this much. So you'll use a graduated cylinder. And again, I will show you what that looks like. And you're going to put 10 milliliters of water in test tube number 1. 10 milliliters of Pepsin in test tube number 2, and you're going to see that it's going to be 10 milliliters of liquid in all 6 of those, but you just have to pay attention to what you are putting in. Number 4, this is where you are going to start filling out your lab report. It says use forceps. Basically, they're like tweezers. Pick up pH test strips and determine the pH for each protein test tube and record your results. I will show you what that looks like. We will run through that. And then you'll write down some predictions in that prediction column on your student guide of what solution will digest the egg white most completely. So here is what your lab report looks like. Your lab report has this chart here. Notice how many points. There's a lot of work involved in doing these pHs and doing that. So we have made this a lot of points so that it's worth your time to actually do the lab. pH level. So you'll put the number and color that you found as the pH level for each of those. And you're going to tell us whether it's an acid, base, or a neutral. Here's where you are going to have maybe a little bit of frustration, mostly because you are not complete with your lab until you wait an entire 24 hours. And then you have to come back into the lab. So once you have completed that first part and you have taken your pHs, you're moving on to part D. And it says place stoppers on top of the test tubes. And once they're covered, you're going to start the timer. The lab will give you a code. You are going to need to either write that down or copy and paste it into a text file. I suggest that you copy and paste it into a text file or maybe onto your lab report sheet. It is the number they gave me was probably about 15 to 20 letters long. So you're going to want to copy that down precisely so that you can get back into your lab. Once you get back into your lab, it will have to be an entire 48 hours after you first started it. So after that 48 hours, that's 2 days. Then you can enter that code back in, and your program will start right back where you left off in your lab. And then part E, we have a lot of stuff crossed out for you. All you are doing is looking at your test tubes, those protein digestion tubes that you set up and you took pH. You're observing those and you're recording those results, your observations, so that you can answer the questions on the lab report. Remember, you must wait 48 hours or 2 days. And then these are the questions that you need to do. You will rank the test tubes. You're actually going to, number one, answer the question, which test tube is the control? Remember that control is something that we didn't change. We changed nothing in that, that test tube to see what we could compare to. We'll rank the test tubes in the greatest amount of digestion and least amount. Use your best judgment. It was difficult for me to see. And then we're going to compare the contents of test tubes 2, 5, and 6, explain which one had most digestion, and then 3, 4, and 6, and explain which one had most digestion. And then in which test tube was the activity, the activity most like that real human stomach, and why do you say that? Once you have that completed, you are then going to upload that into the Dropbox. Dropbox looks like this, and that is going to be due by Monday, March 20th. So we're starting the lab on Friday. You're going to wait 2 days, so 2 days, depending on when you started that lab, could potentially be Sunday. And then by Monday, you should be able to answer those, get back into your lab, answer those questions, and upload that lab back into that Dropbox by 11:59 p.m. So that means that you are making sure you are starting the lab on Friday. If you don't start it on Friday, you will not be able to complete it on Monday. So here are those announcements for when you start the lab. Make sure you start that lab Friday, March 17th. Copy down that code so that you can re-enter the lab 48 hours later. And that help session is going to be Friday, March 17th as well. And then the lab will be due Monday, March 20th. So here is how we are entering into that lab. So first of all, I'm going here and I am going to download. I'm going to read this and read the objectives, and I'll download this student guide. Once I have that downloaded, you can print it off. It would be easiest if it was printed off. And then I'm going to read this information here. Make sure you go through all of this and look at the process of digestion as well. And looking at your pH as well. Once you've done that, you are ready to enter into that lab. Couple safety things. And here is your lab right here. Human Digestion Virtual Lab. Once you are here, you're going to view the tutorial. It'll ask you to pick gloves, and it's going to walk you through some information. So I'm going to start the lab. It says place your mouse over the 4 arrows. If I move these arrows, it moves up and down. This is how you maneuver around a lot of this lab. You're going to need to use all of these arrows. Look down. Here's your forceps. The gear hides that menu. I like to keep my menu open. I can resize. So I can go like this, and it changed the size of things. That lock is going to turn off or on that scrolling. So if you are working on something over here and you don't want the screen to move, you can go like this and it locks it so you cannot move the screen. Just push it back in and click the speaker icon to turn the lab audio on or off right there. And then the drag bar to magnify